Thank you. So, is this on now? Okay, good. All right. As you see from the title, estrogen, sewage, and sea anemones. I want you to leave tonight thinking about the types of human-generated pollution and the impact it is on the environment. So that's your goal. And now I'm going to kind of walk you through how I'm going to present you with material to kind of illustrate and demonstrate how human-generated pollution, a term we oftentimes use with this is anthropogenic pollution, human-generated pollution, and see what this does with anemones. Now, I suspect for most of you, if my little clicker will work, maybe it's not going to do that now, so I'll have to do this. Is there anybody in this room that doesn't know what that's to? I mean, what movie that's to? I mean, I think, how many know what movie that's to? Okay, yeah, most everybody knows Finding Nemo. Did you ever think that Finding Nemo was telling you some biologically accurate information? Well, it is. This scene in particular is the scene that I wanted to show you because this is real life. It's not just the little talking fish. They're on a pipe. These pipes really do occur naturally. I say naturally. They're put out there in the real world. The pipes are actually laid out at most coastal cities. This is a sewage outfall. Now, you remember from the movie, Nemo goes down the pipe in the dentist's office, and then he's, washed out. Well, there really are sewage outfalls at coastal cities, and typically most coastal cities have a designated distance and depth that outfalls need to go before they can be discharged. Now, to be discharged, they're going to have to, go out and literally just shoot it out through the pipe. So here is a sewage outfall in Florida. These things do exist. Now, on this picture, I want you to notice out at the edge of the plume over there, you see a whole little school of fish. Okay? Now, this is not a cannon shooting feces out into the ocean. This is secondary treated effluent. It's nutrient enriched. There's a lot of nutrients, and that's why you see the school of fish out there. And they actually have to manage. The states and the governments will look to manage how this sewage effluent is sent out into the ocean. They don't want just untreated sewage, and so there's the sewage treatment plants, and all of that process is involved with discharging this treated sewage effluent out into the ocean. Because obviously it's highly concentrated right here. But as water, high concentration, your chemistry, diffusion, high concentration to low concentration, it'll just separate out. So the issue is, what is coming out of that pipe? Okay? It's not just feces and urine. There's a lot of other stuff. And so what I want to do tonight is kind of walk you through a little bit of that. And so I'm going to give you a few terms to think out. Here's the first one. Persistent organic pollutants, POPs. Okay? So what are we talking about with a POP? We're talking about chemicals. We're talking about human-generated chemicals that you see are present in the environment. They tend to stay there a long time, and they tend to get dispersed by wind and water. They don't break down. They tend to be present and detectable for a long period of time. And you see the EPA describes POPs as a global concern. Now, something you probably didn't think about it quite as a POP, but yet it still is related. We've all been to some place and seen plastics. and you've seen them accumulate. I can go to these remote islands down in the Bahamas that nobody lives on. There's never been a house in the recent history anyhow, and there will be plastics on the seashore. So plastics are everywhere. But not just plastics are everywhere. They leach and chemicals seep out of them, okay? So those are what I'm referring to as those, POPs. This is a picture that I pulled this off of the web just recently. You can see here there are places, especially out in the Pacific, where you've got these large gyres that the ocean currents are literally collecting just these large spans of stuff that floats, a lot of plastics. I can go out in the middle of the deep ocean and find plastic jugs floating. And there'll be some little fish that'll float right under, be sitting there right under the plastic that's floating. Pallets, wooden pallets floating. So there can be all kinds of stuff. And so you can see here that the amount of plastics that are accumulating in the ocean is huge. 270 tons is what they expected back in 2014. So this is a big deal. This is a lot of material. And I want you to think about all this material as it's releasing other chemicals. Okay? There are chemicals that get released from plastics. One group is called a phthalate. And so these are out there. Now, what do these things tend to do? Well, I put up here some examples of some articles that have been published in different places. In this case, there are those of you who are probably from Florida. There's probably anybody from Florida? Okay. Those who in Central Florida, there's Lake Apopka in Central Florida. And it's been well documented, decades worth, that the alligator population in Central Florida was, for a while, had an unusual balance of males to females. It wasn't a 50-50. Now, normally alligators take and they create this nest, and the female lays the eggs, and the eggs don't have a predetermined sex when they're laid. Initially, they're laid, and depending upon the temperature, they either develop into males or they develop into females. And there's a critical threshold where, and in the nest, there's a difference in temperature. So I think it's the lower ones are the males. I can't remember the switches. And then a higher temperature is the opposite sex. Well, they started noticing that there's not an even distribution of the males versus females in Apopka. And they end up, as they started to look at this closer, they discovered there were these chemicals that were getting washed in. Now, all around Lake Apopka, those who live down there know, got a lot of agriculture. And anytime you have agriculture, what kind of chemicals do you want to use to protect your crops? Pesticides, herbicides, or you want to keep the insecticides to keep down the insects. You want herbicides to keep down the weeds. Well, you also are familiar in Florida with having rain, downpours at times, and all that stuff washes off, collects in the lakes. So maybe the farmers planted, sprayed it, distributed the stuff over their crops, but then it washes off the crops and down into the lakes. And so it had an effect. It had an environmental effect on the alligators. Here's another example of a pesticide that cause males to start acting like females. And in this case, there's a picture of two male frogs that are mating. When amphibians mate, it's called amplexus, and under the normal circumstances, that the larger female is the male's on her back and basically gives the Heimlich. She releases the eggs, he releases the sperm. It's all an external fertilization, and that's how the eggs then are fertilized and laid. But in the presence of a pesticide, there's evidence of males turning into females, and they produce eggs, but they produce eggs that instead of having an X and a Y chromosome, they have two Y chromosomes. So this is another example of chemicals that are tweaking development of organisms, okay? So I wanted to kind of walk you through a little bit more of one other idea, and that is now human pharmaceuticals. That big pipe that I showed you, that sewage outfall, also has coming out of it human pharmaceuticals. Your grandma takes blood pressure medicine. It's showing up. You know, you got an uncle who takes statins to control their cholesterol. They're showing up. You're taking NSAIDs, non-steroidal anti-inflammatory drugs. They're showing up. You can go out to those sewage outfalls and start to test the water, and you can find all the different types of classes of human pharmaceuticals. One of the most common ones you'll find is estrogen. Estrogen, you can actually detect the difference between natural estrogen and synthetic estrogen. Synthetic estrogen, the source of that is typically birth control pills. So here you have chemicals, all these human pharmaceuticals, you have chemicals that are actually ingested through a human, goes through our whole digestive tract, excreted, going all the way through a sewage treatment plant, and still these chemicals are making it out to the outside of that outfall intact. They're making it without getting completely broken down all the way through that system. Okay? So with that in mind, that's why I'm starting to look at these kinds of things, because we know what are the targets of these chemicals. With these human pharmaceuticals, you take a certain drug because you want it to have a certain effect in your health. Okay? It has a certain tissue that it targets. You know, if you have a headache, you take the aspirin. You can even detect, even when it comes to chemicals that show up in the outfalls, even when it's not necessarily for a therapeutic value. Another chemical that makes it, caffeine. You know, if you drink caffeine, you can detect caffeine in these outfalls. Okay? So that is, with this kind of the background information, I show you this paper because it was this paper, when I first saw this a number of years ago, that started getting me to thinking about this. And it's not only me who's thought about this. You can see here, this paper has been cited over 2,000 times since it was originally produced in 2006. So a lot of people have started to pay a lot of close attention as to what is this that's coming out of these outfalls, and what impact can these chemicals have on animals in the environment. Okay? So that leads us to this next idea, endocrine disruption. What about the possibility that some of these chemicals, like estrogen, if it shows up out there, estrogen works on mammals, but there's other animals who also have sex hormones. Well, if you have an exogenous amount of these hormones and they're floating out into the environment, maybe these chemicals are having an impact on other animals out in the environment. So that's the idea behind endocrine disruption, which will actually cause impacts with development. So if these chemicals are just seeping out there, maybe they're impacting the reproduction, the development, and you see the way neurons are functioning, the way their immune systems work, they can disrupt that. So when we start talking about things like estrogen, testosterone, you've all heard the stories about, you know, what kinds of chemicals do world athletes try to take and get an edge, and they're usually considered illegal? Anabolic steroids. Okay, those are sterols. Those chemicals are intended to help build muscle mass. Okay, so these chemicals can influence changes in development. Okay? So you can see here these sterols are also considered some of these persistent organic pollutants. They're large organic complex molecules, and their concentrations are quite small. Quite small. You see here 1 nanogram per liter. That is a part per trillion. Okay? 2 micrograms per liter. 2 parts per billion. Okay? Small numbers. Okay? We know that, you know, the Olympic athletes, when they take you know, they're competing, they get a urine test when they're done. And part of the reason why they get a urine test is they detect for any kind of enhancing drugs, and because excessive amounts will be excreted. All right? So, what I study is corals and sea anemones. And this whole idea of endocrine disruption has been very difficult to detect in corals. Okay? It's been pretty well documented in a number of different vertebrates. For example, fish. They've seen evidence of endocrine disruption in fish. I just showed you the pictures of the frogs. That is another form of endocrine disruption in another vertebrate. So frogs and fish and mammals and birds, you can see evidence of these types of things. Okay? The Fent paper that I referenced a few minutes ago, one of the opening examples they give there was in India there were farms that were giving some drugs to the cows, the cattle, to help them. They had I think it was a flu or something. There was some kind of a livestock disease, so they were given some medication, and then some of the cattle died, and then after the cattle died, vultures would come and eat the carcasses of the cattle, and it turns out it nearly decimated the vulture population because those chemicals that weren't harmful to the cattle were actually killing the birds. Okay, so these kinds of things are pretty well identified in vertebrates, but in an invertebrate like a coral and a sea anemone, this isn't so well known. So that was what kind of had my attention. Now, this kind of illustrates this idea of endocrine disruption, kind of as an illustration. You can see here, as our populations grow, there's an increased consumption of various types of medications and steroids. That's going to increase the effluents that is going to go through the sewage treatment plants. And then you run the risk of, if you start to have changes with where they live and if there's any environmental changes, you know, typically in rivers, you know, I say you as in, you know, the United States, we've changed the whole flow of the Colorado River. There's hardly anything the Colorado River hardly flows into the Gulf of California anymore. It's because it's pulled off for agricultural purposes all along its shoreline. So, you know, there can be environmental changes. And then you throw in, you know, the Colorado River that's rolling through the Grand Canyon isn't the same temperature as the Colorado River would have been if it hadn't been dammed. Okay? So as the environment gets manipulated, there are these changes that can occur. So that is kind of illustrated kind of by this idea here. You see, you've got 1, 2, 3, 4, 5, 6. These are all different kinds of places that are related to the endocrine disruption. Take your number 1 over there. It's our own homes. Okay? How do we generate pollution in our homes? Well, one of the ways is some of our homes are on septic tanks, not on city sewer systems. And septic tanks in the Florida Keys, they just had the big hurricane that blew through there. In the Florida Keys, you've got 100 miles of islands, and you don't have a municipal sewer system. There's not a bunch of pipes going between all these islands. Everybody's on their own private little septic tank, okay? But the ground in the Florida Keys is primarily calcium carbonate. That kind of porous rock material allows things to seep through it, okay? You look at the number, so that's the number 2 is representing septic tanks. And of course, septic tanks then, the materials can leach out of there and even go down into aquifers. So you can get into aquifers. Now you have a large pool of water that could be potentially contaminated or be distributed other places. Number 3, hospitals. Hospitals, of course, are where you have lots of pharmaceuticals. And the regulations have changed, but there used to be a time when if you had old medicines at the hospital, you disposed of it by washing it down the drain. Now, they don't do that anymore, but those are things that have occurred in the past. Now you have your sewage treatment plants. Number 4, you have industries that generate chemicals. Paper mills. Here in the southeast, making paper, you produce chemicals, and you need a lot of water. You could be producing these chemicals, and once again, these persistent organic pollutants. Farms. What kind of livestock are typically on farms? Mammals, pigs, cows, sheep. All of those mammals also have menstrual cycles. So all those same sex hormones are getting released from those places as well. And then, of course, you can see that from the sewage plant, you have the sewage outfall, so you pump the treated sewage out in the ocean, but some of it may also go down into, like I said, the aquifers and that type of thing. I show you these 2 pictures. These are some aquifers that are down in the Yucatan Peninsula. It looks pretty cool. They've done some water chemistry down there too, especially under some of the places you've got aquifers down underneath some big resorts. There's resort towns there in the Yucatan Peninsula. You know what they've detected in this water, in these aquifers underneath there? Cocaine. There is enough cocaine that is getting urinated and into these septic tanks on these resort cities, and it's leaking through the calcium carbonate rock, and they can detect it in the aquifers. All right. So this whole idea is what the human sewage, and the way it started getting my attention working with corals is some people that I know actually looked at in the Keys, they discovered that you can see this coral with these white spots here. This is called a white pox disease, and it turns out this disease in the coral in the Keys is caused by bacteria that is known to come from human sewage. So people aren't going out and going to the bathroom out on the reefs. What they're doing is they're using their toilets in their homes with the septic tanks that are built to code, and yet the chemicals are leaking out. In this case, bacteria. This is a particular bacteria called Serratia marcescens, and it is known to be the agent that causes this particular coral disease. So, what I want to show you now is, here are 3 different papers that kind of have been done in the past, 10 years ago or a little even longer, detected estrogen in sewage in the coastal marine environments. They've detected steroids and hormones in South Florida. And you can see the bottom one then is by a woman named Anne Tarrant that about 10 years ago, she kind of threw up her hands. She had been looking at hormones and cnidarians and corals and sea anemones, and she wrote this paper 10 years ago going, hormonal signaling in cnidarians, do we understand the pathways well enough to know if they're being disrupted? She could not figure out how, if there are these hormones, and it turns out that cnidarians do have these sex hormones, but this whole idea of endocrine disruption didn't really seem to be working. And as time has gone by, we have gotten sequence data. We've got sequencing data for the genomes of some cnidarians. And it turns out that when we've sequenced the genomes of corals and sea anemones, there's one particular protein that corals don't have. You see these little green illustrations. Those are those estrogen receptors. Now, from your general biology, those who are in or have had, you know that your plasma membrane is a barrier. And as a barrier, it doesn't allow charged molecules and polar molecules through. But nonpolar, hydrophobic molecules, such as a steroid, can move through and is not inhibited by the membrane. So what happens is those large sterols, steroid hormones, move through and bind to a receptor that actually can go through not just the plasma membrane, but can go through the nuclear membrane and can bind into these proteins that are in the nucleus. It's called an estrogen receptor. Well, it turns out cnidarians don't have them. Cnidarians don't have any of them. So when Ann Tarrant says, I don't know what's going on there, it's because the sequencing data says, how can you have endocrine disruption if you don't have those little proteins that when they bind to estrogen, you see after they bind to estrogen, you can see over here, then they bind to the DNA. And then when they bind to the DNA, they change which genes are being turned on and turned off. All right? So without that critical protein, there were people who just kind of threw up their arms and go, well, endocrine disruption, yeah, I don't know if that really occurs here. Because if they had that, then see, estrogen and those signals stimulate cell division. If it was an anabolic steroid, it stimulates cell division of muscle cells, and so those people, guys, athletes take it and get more muscle. But there's also changes in cell division and gene expression if estrogen binds. And so suddenly, but these signals, these critical pieces of the puzzle are not present in the cnidarians. So that's what I used as my backdrop. Okay, there are these signals, they're out there, and they're shooting these outfalls out to where corals are at. Why can't we detect this? Why can't we detect this? So what I did is I started to do a very focused little project where I took some sea anemones and I exposed them to 17-beta estradiol. This is the most potent form of estrogen. Okay? And what I did is I exposed them for 3 different concentrations. And I did it for 4 hours. Not long. I just gave them 4-hour dose. That's 0.2 micrograms in a liter, or 2 micrograms in a liter, or 20 micrograms in a liter. So I took 3 different concentrations, magnitude of order difference, 4 hours. That's all I did. Okay? From there, what I did is I took some other anemones and I exposed it to some different things. So not only to estradiol, I exposed him to testosterone, another hormone. I exposed him to cholesterol. Why do you think cholesterol would have been I chose to do cholesterol? Anybody want to take a wild hair guess? Think about what you're learning in membranes, in your introductory bio class. Cholesterol is in the membrane, isn't it? So I wanted just to expose them to the same kind of chemical that influences the fluidity of their membranes. I exposed them to naphthalene. Now, those who may have had or are in or will be soon organic chemistry, aromatic rings, 2 benzene rings put together are naphthalene. And it also represents the simplest form of a polyaromatic hydrocarbon, which is also one of the smallest forms of a component in crude oil. In addition to the straight-chained hydrocarbons, you've got these cyclic aromatic structures. So that's one of the simplest one of those. So I exposed him to that. And then lastly, I exposed him to a pesticide, an organochlorine pesticide. So a lot of pesticides have a lot of chlorine atoms stuck around. So I exposed him to these different chemicals. And I also then took, with the group that I exposed to 20 parts per billion of estrogen, estradiol, after I gave them 4 hours with the exposure, I pulled them out of there and put them back in nice fresh water. Not much different than if you're stuck in a room where everybody's smoking and you want to get a breather, so you go outside and you get fresh air, okay? So I put them back in fresh water. And then, after they've been back in fresh clean seawater, after they've been in there 4 hours, I extracted some of the tissues from them. I took some others, let them go 24 hours, then I extracted tissues 96 hours, which is 3 days, 4 days, took those tissues out, 7 days, took them all out. So I wanted to see how quickly could they recover from the exposures to these different things. And then, of course, after 7 days, I still had some. I said, okay, I'm going to give them a second pulse of estradiol, and see, and then extract those tissues. And then what I did is I extracted the RNAs, the messenger RNAs. So up to this point, everything you've heard me talk about kind of sounds like environmental, ecological kind of stuff, maybe a little developmental. Well, it's got a pretty significant molecular component to it too. So now your understanding of messenger RNA, transcription and translation, all those kind of things start to play a role. So I use this kind of a technique where I took advantage of characteristics of mRNAs, and I isolated them because I wanted to see, are there genes that are turned on in exposure to estradiol? I want to find out, are there genes that are uniquely, and I think of it almost like a fingerprint. Can I find evidence of an exposure if I wasn't there to see the exposure happen? And so that's what I was looking for by doing this work. Now, after I got done, I actually isolated 360 fragments of different mRNAs. And when I started to compare the sequence information, and I used publicly available online databases at GenBank, the National Center, for Bioinformation, I don't remember, NCBI is the abbreviation I call. I was able to identify that I actually had some genes that were associated with estrogen. I got something that maybe could be of value here. So that's what that showed me. So then what I needed to do is, now if these genes are associated with exposures to estrogen, well, now I need to know, can I find them in different levels? Is the level of an mRNA that's expressed in that 20 ppb of estradiol, is it similar or different from the one that was 2 ppb or 0.2 ppb? So I did that, and I was wanting to see things like dose response, how quickly they could recover, what happens if they get that second exposure. And then I also, as I told you, I looked at different kinds of chemicals. And so looking at all these different, I wanted to see, can I find evidence almost like the criminal that leaves the fingerprint, but he's no longer there. So you know somebody stole the truck, but you don't know who it is, but you got a fingerprint. Okay? So that's what I'm wanting to look for. Turns out I found 13 genes. I found others, but these 13 really stood out. Now I'm not going to expect you to memorize this whole list, but let me walk you through a couple quick features. The very first gene, 17-beta hydroxy steroid dehydrogenase, probably doesn't mean anything to you, but let me tell you why you should want to know about it. It's the last enzyme in the pathway for synthesizing estrogen. So here, I just expose them to estradiol, the most potent form of estrogen, and the first mRNA that I find represents the very last enzyme in the estrogen synthesis pathway. I said, hey, I must be on the right track to doing something. If I'm getting something transcribed as a result of my exposing these animals to this chemical. Then I started noticing I got an immune response. I got an inflammatory response. I got chemicals that were associated with breaking apart vacuoles, and I got chemicals that were associated with binding sterols. That Niemann-Pick, number 6. That Niemann-Pick number that I have there at number 6, it is known to bind a sterol. I was using estrogen as a sterol. Estradiol is a sterol. I had another gene there, number 7, placenta-specific. I'm going, I don't know what this is, but the name is telling me I better pay attention to this, so I start looking at this. And then I noticed, number 9, I had something called a sterol pump. Number 10, involved with sterol binding. Are you getting the clues that I'm seeing? I'm getting, as I looked into these things, I'm finding a number of different genes that have some connection to sterols. So, with that, I needed to quantify this stuff. I needed to now, I did a type of molecular assay known as real-time quantitative PCR. It is a highly sensitive technique that I can actually calculate and quantify the amount of an mRNA, a particular mRNA that's expressed in one set of tissues, how it is relative to another tissue. So I use that very sensitive technique to kind of look at these 13 genes. And what I discovered is, here's those first 3. Notice in this case, this is that dose response. Notice that my control samples, 0 ppb. These genes aren't turned on. They're not turned on at all. 2 ppb, 0.22, and you see a trend. It increases its concentration. So we've got a clear relationship here with dose response. Some genes are clearly responding to the presence of it and the amount of it. Okay? So I said, okay, well, let's take a closer look at some of these other ones. Now, these next 3 genes also have a relationship. Vesicles that come, when a cell forms a vesicle through endocytosis. Turns out cathepsin is a lysosomal protease. It's in lysosomes, which are those organelles that digest things. And when the lysosome fuses with a vesicle that's coming in through endocytosis, it can digest the interior of what's in that vesicle. Turns out that NPC is that Niemann-Pick, which is a protein that's binding the sterol. And the red, the equistatin, is a regulator of cathepsin. So it's kind of like you got something here for gas, pedal, and brake. Which one's driving the process here? And you see there's some differences between the concentrations, but generally speaking, the dose response isn't quite as evident with these, but those were 3 that were together that were of interest. Now, I hit one too many here, so let me go back up 1. Oops, I hit the wrong thing. Turns out these next 2, there's that placenta-specific and another little transcription factor. And the blue line, the placenta-specific, is a regulator of that other transcription factor. So these are connected. They have an interaction between the 2 of those. The next 3 that I want to show you, these 3 are actually involved. These three proteins here, these 3 genes, are actually involved with a developmental pathway known as hedgehog signaling. And hedgehog signaling involves cholesterol, which is a sterol also. So different concentrations of estrogen is tweaking a cholesterol-mediated developmental pathway. All right? Now, after I looked at these 2, backed up. These jumped out at me. This is a gene, the purplish colored one here is a gene known as vitellogen. It's an egg yolk protein. It's associated with the development of eggs. concentration is too high, this gene and this last gene, which is called gametogenesis, is also associated with cell division and development of gametes. Okay? So I had clues now that said, okay, what else have I got here? So I'm looking at these genes now, and I looked at the recovery, the same set of genes, okay? And so here were 4 genes that I want to show you. So there's the enzyme that metabolizes the final step of the estrogen pathway. And then there was this plaquate, this placenta-specific, and it did something very different. But like I pointed out before, plaquate and the CEBB, that enhancer binding protein, the third one over there, they're related as far as that the plaquate regulates this third one. And this third one doesn't look strikingly similar in the pattern of as you go through the controls. You notice that none of the control anemones expressed any of these genes. When you gave them the exposures, you can see that the black squares are where the exposure, where they got 4 hours of 20 ppb estradiol. And you see they deviated from the control. And then over a week, when they go through the periods of recovery, they are still elevated. Notice that a week later, they're still the hydroxy, the 17-beta and the CEBP. You see, they're still elevated. They're still significantly elevated. And in the course of a week. So the signal was gone, and at 7 days, they're still doing this stuff. So it's kind of like, evidence that the arsonist was there. Okay? And then, of course, the last gene was really different. Here is that telegenic, here's that egg protein. And notice that early on, it didn't change much compared to the controls didn't. Even the first exposure didn't do it. But 4 hours after the first exposure, when it's back in fresh seawater, it's kicking up and it stays elevated for the rest of the time. So I have these clues once again that there's evidence that once these chemicals have been exposed, these anemones have exposed these chemicals, there's changes in which genes are turned on and how long they're on. Okay? A couple of the others, there's 2 genes that are involved with immune responses and inflammation. And in this case, you see, they initially, when they get their first exposure, jumps up. But as time goes by, it declines back down. And by the time they get their second exposure, it's kind of like, yeah, we've seen that before. It's nothing new. Okay. So they're clustered in these responses. Now these next 3 were the ones who were all involved with that signaling, that developmental signaling involving cholesterol transport. And they're showing a general trend. Wherever their first exposure, and you see that a week later, second exposure, it's all they're reducing. So this suite of genes is different from when they were first exposed. Remember that the first data point in all these is your control that isn't expressing these. These are not operating. There doesn't mean there's an absence, but there is a basal level. It's not radically up and down regulated. Now, the final one are these 3 genes that are involved with those vesicles that are getting transported in by endocytosis. Phagocytosis you hear about too. So as these vesicles are coming in, we see that the relationships here. Notice that the protease and So you see the naphthalene and the lindane, real big difference. All right, now the next three. There's some differences here. As far as whether cathepsin is driving the process or equistatin is driving the process, and Niemann Pick. I want to just focus on the estradiol one. You see that equistatin is upregulated, which means it's going to stop the cathepsin, which is also going to stop, if cathepsin is no longer working, then Niemann Pick, the green one, is going to not be, you don't want to be bringing more of it in if you don't want to have to metabolize it. So there's some difference here, and now you're starting to see these other things show up over here with the naphthalene and the lindane as well. The next two were the two that were transcription factors, and the plaque was the one that regulated the EPB. Okay? And so in every case, they all did the same things. Plaque is down, and the EPB is up. Okay? Now, the next three. These are the three that are involved with that developmental pathway that uses cholesterol, the hedgehog signaler. And you see, cholesterol and testosterone look quite similar. Naphthalene is looking really weird. And you see a similar response with naphthalene, and there's not really much, way of significant response. But notice your controls. None of these are getting expressed significantly in the controls at all. So all of these genes are getting turned on. If they're being upregulated, they're above this zero line. If they're getting downregulated, it's below the zero line. So some of these, they're wanting to get more of it. Others, no, we don't want it anymore. We'll back it off. Okay? Now there's a big difference. Vitellogenin, that egg yolk protein, and gametogenetin, that is also involved with cell division, and you see a huge difference here. Okay? So what you're looking at is if you look at the suite of these 13 genes, you're seeing how you can now, if you take this suite of 13 and somebody says, here's an anemone, has it been exposed? You could conceivably tell whether it's been exposed to estradiol, testosterone, cholesterol, naphthalene, if they were exposed to one of them at 20 parts per billion. You can start to differentiate. So in a sense, you've seen those programs, Forensic Files, NCIS, and all those guys that do all that molecular stuff, but they seem to do it within 45 minutes of a show. Okay? You don't do it that fast, but it can be done. Okay? And the information that you can get is pretty powerful. All right. I show you this picture just temporarily because there's the visualization of how a vesicle is brought into the cell. And you can see up there the LDL. This is basically the pathway that's used by if you get your blood drawn and they check you for good cholesterol, bad cholesterol, you have LDLs, low density lipoproteins. Okay? This illustrates that. And you can see here that cholesterol can't go through those cells because that long table. It's so hydrophobic, you can't get cholesterol inside the membrane. It's going to stick in the membrane. It's not going to go through it. It's going to stick in it. So to get cholesterol inside the cell, it has to go in with a low-density lipoprotein, LDL, or high-density lipoprotein. So once you bring it in and you form those once you bring in those vesicles that you see, I can't bring up my pointer on there. So what you have to do then is you have to use your lysosomes and digest it and separate the cholesterol from the proteins. And then you can release the cholesterol into the cell And then this Dehydrogenase, the last gene I wanted to show you some big signals. I put that in this corner. Here is the structure of dioxin, another chemical, some hydrocarbons and some oxygen. And you'll see that dioxin induces and it interferes with spermatogenesis, another gamete formation and cell division. So, I looked at just these three genes and I discovered something this summer. This summer, I taught a special course out at Rosario. And so, when I was out there at Rosario this summer, I had some students do some work with these three genes. And their work was great. And so, since the genes work well out there, I brought them back to Barry, and I had my student workers back there at Barry start to use them. And when they started to use them back here at Barry, they didn't work. I'm going, these things, and I mean, the people I have working on it Barry are experienced, and this wasn't working. And I'm going, I was double back and looking at what I had from the summer, and it was beautiful work. I mean, the genes were working good for this qPCR stuff, but they weren't working this fall. And so I went back, and I started looking at just anemone tissues that I had sampled at different times. And I compared the anemone tissue that I had sampled in the spring from the anemone tissue that I sampled in the summer. And you can see here, these three genes didn't amplify. In fact, we're talking, these genes, the vitellogen and the Dehydrogenase, were absolutely not being transcribed at all in the spring. What I discovered is the reproductive stage of the summer, these anemones that were taken during the summer were in the process of developing their own gametes. They were getting ready to spawn. So since they were getting ready to spawn, these genes are high. These anemones here were not exposed to anemones. Okay? So this is the information that I discovered as I finished up the summer. So what you're seeing here tonight is evidence of endocrine disruption in an animal that hasn't previously had it documented. I can't get this out fast enough. So I think I'll stop with that. But take a question or